summer school. Thank you very much for waking up yet again and coming to this talk. As Professor Takachi said, my name is Sunčica Zdravković. I work in Novi Sad at the psychology department. And actually, I was asked to this project because what I do is visual perception. And visual perception is something I've always found fascinating. So my main topic is not how to teach children, but how do we acquire information. And how do we acquire information through one extremely sophisticated and well-developed sense, visual sense. So I'll, of course, start with the trick question, which is, how do we see? Have you ever thought about that? And you can probably tell me, we open our eyes and we see the world around us. Yes? But if I offer you something like this, what do you see here? This is not a trick question. Buildings. Buildings, lovely. What else? Water. Sky. Reflections. Does anybody see night? Lovely. Here we are getting to things. Does anybody see a city?
Yes.
Do you know where this city could be? Is it a village in Netherlands?
No.
What is it? Shanghai, somebody actually knows. Yes, it's city far east, obviously. Yes, so we can see these things. So when I offer you an image that I suspect you, or most of you, have never seen before, you just blink, and 120 milliseconds later, you see buildings, waters, reflection, and somebody even saw Shanghai, right? Effortlessly. All 75 of you sitting here did this immediately, and without actual effort, like, how do I do this? Colors, shapes, background, time of the day. No, it came to you immediately. So this is what I mean when I say, how do we see things around us? And actually, it is quite effortless and instantaneous to us to gather information in this way. But this involves a huge portion of the brain. It has, I can demonstrate you a lot of amazing phenomena of normal vision, distinctive pathological casing, and difficulties when we want to engineer this process or model this process. Amazing phenomena of normal visions include, among other things, illusions. So I'm just going to show you examples that I'm sure you are very familiar with. This is a lovely capture wall illusion, and these lines here are parallel. Hopefully you can't possibly see them as parallel, right? Yeah? These red balls are of the same size. Again, it is very difficult to see them that way. And I can help you a bit to see that things are exactly as I'm saying by changing the context or relevant context a bit. So you can see illusions easily. It gets even better. You probably are familiar with these things that are called ambiguous figure. So a duck or a rabbit, right? You can see both things depending if you start. Huh?
Rabbit or a bird?
Well, duck. It's a bird. Okay. So it's either a duck with an eye, if you look from that side. If you start with this being rabbit ears, eye again, and the rabbit nose, right? You can see two things at the same time. So ambiguous images. Can you see them?
Yes.
Excellent. And then, loveliest of all, impossible objects and scenes, and you've seen a lot of those as well. So all of these you can find a lot of examples on the internet, so I'm not going to need to tell you. But what is lovely here, in images like this, locally they are perfectly fine. But globally, there is a problem. This problem is of course achieved by making mischief when transferring 3D into 2D, but there are other ways to achieve these things. However, what is very interesting for me when you look at these objects is that your brain does not complain. It produces a percept. You can see this. And it's only when I start telling you, oh, look at here, then you start actually seeing the problem. But before you looked at it, and it was fine. And you can see all of these creatures from legends and everything else we show you, and your brain will take information from outside world and create a percept for you. Very good, very reliable, very fast percept. done? Well, one of the things that clues us in are pathological cases. There is a patient with this interactive agnosia, and when asked by the doctor to do a drawing of a human figure, this is how you end up. Now, this is even obvious to us amateurs that this is not a problem of not having the skills to draw, right? Because even if you don't have skills to draw, ear or nose won't end up outside of the face. So this is telling us when this complicated sense falls apart, what happens and how it falls apart. And this helps us model how it actually works. And of course, why do we want to model? Well, we would like to see, we would like to see if you take a complex picture like this that all of you can see nicely, and you take a part of this picture, and you take it pixel by pixel, this is how you end up. Now, you want to have a machine that will tell you, look, this is an illumination difference. This is where the sun spotlight hits the floor, and this is where the girl is standing, and this is why this picture is taken, because to us it gives us aesthetic pleasure. But this aesthetic pleasure includes that we understand that this is a spotlight, and this is a change of color when it comes between the window and the wall, right? I want my machine to be able to do the same for various reasons. If I send a little robot to Mars to walk around the neighborhood and pick up samples, I want it not to stop when it gets to the shadow, but I want it to stop before it falls from the cliff. So I want this robot to be able to clearly distinguish the situation of the cliff and of the shadow. And we do that effortlessly, and I don't want my robot to have to call me every time it ends up with a shadow or a slightly strange terrain, to call me 12 minutes one way, 12 minutes the other way, till I tell, no, don't worry, it's a shadow, keep moving, right? I want to be able to model this. And then when I'm able to model this, I can tell you, okay, I understand how this information process is taking place. How we acquire information, how that information is processed by the brain, and then how these percepts do emerge. These useful percepts that help me not bump into things every time I move around. And even if these things are not stationary, but when you're at a crowded party and you move among people, you don't bump into people unless you're helped by alcohol. Okay. So this is what I mean when I say, how do we see when we open our eyes? How this information is acquired. And this amazing sense, vision sense, did not have a history like other senses. If you take geological time, and if you kind of measure the efficiency of the sense, then other senses slowly went higher up in efficiency over the time. So it wasn't exactly a linear process, but they were getting there slowly. However, about 543 million years ago, there were 38 families of living things inhabiting the earth. And one morning, 6 of them woke up and decided to invest a huge amount of metabolic energy. Basically, in the case of beets, 80% of everything they eat goes into vision. It's a huge, huge investment. Those 6 families decided, okay, we'll make this investment for our offspring. And they did well, because they rapidly developed a new, amazing sense called vision, and they ended up having 95% of biomass today on Earth coming from these 6 families. Developing, well, most wonderful eyes you have ever seen on this planet, obviously. Very different engineering solutions for us to think about, but always with the same function, to gather the information from light and to actually transfer it into meaningful percepts. This abrupt evolution of vision was actually confusing Darwin itself. He couldn't understand how a sense so complex and so fantastic could develop so fast in geological time. Today we actually have some models that explain how this is happening. In less than 100 million generations, It's quite remarkable, actually. So the stimuli for this sense is light, or actually carrier for the information is light. And let me just remind you about the things you know already about the light. It's an electromagnetic emission. It's a complex wavelength because it has electro and magnetic component, and they are perpendicular to one another. And physicists nowadays have two descriptions for light, as a particle and as a wave. But in optics, traditionally, and therefore in visual perception, we use the description as a wave. So that's how we do it. But it can be done both ways in physics. And of course, we don't see the full portion of electromagnetic radiation, but only a small proportion of it. So vision is sensitive only to a small proportion of it, between, let's say, 380 and 750 nanometers. You'll find different measures in different books. Don't worry about that. It depends on the way it was measured and the sample and so forth. But this is actually a range. So, back to how do we see? So we see objects and events around us. So not just objects and static things. Right now, you are perceiving my talk. It's an event, right? So this desk is object, but you can perceive events. So also, there is a time component to this. Light carries this information, and what we evolved to see is basically reflected light. So it comes from a light source, it reflects, and carries information about an object. That's what we are evolved to see. Obviously, nowadays, you spend a lot of time staring at light sources. You know some, of course. Computer screens, mobile devices, this is not reflected light. But generally, we are talking about reflected light. So light reflects from a light source, comes to the objects. It changes after being reflected, and then comes to this visual system. The first thing is an eye, and you've seen an eye, and I'm going to show you a plastic model of an eye. So this is a plastic model, don't get alarmed. And this plastic model shows how eyes look inside. And inside this white ball is what really interests us, and this is a layer called retina that has these special cells. These are neurons, and these neurons are photoreceptors. They're cell receptive or sensitive to light, henceforth, right? Photoreceptors. So you actually have this in artificial devices as well. These photoreceptors have a simple mechanism. They have some chemical called opsin, and when that opsin gets torn down by incoming light, it sends electrical signals. These are neural signals that we later progress through our brain. Okay, and we distinguish two types of these cells based on their intensity. These rods, which say night vision, are very, very sensitive to extremely low levels of light. Good control experiments actually measure that 1 quantum of light is already enough for a human vision. Not in these conditions, but in well-done condition when a person is adapted to dark and so forth. So one quantum of light. So we're talking about the extremely precise device for detecting light. And cones differ in quality. So you have 3 types of the cones. And here are the graphs that you might have had a chance of seeing. Here we have different wavelengths. And here we have the relative absorbance. And these are those 3 types of our cones. Now, usually old and wrong biology textbooks would tell you that you have blue, green, and red cones. Please, 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 never again, after this talk, refer to cones in that way. The cones are long waves, middle, and short. And this is extremely important for me that you understand for the future modeling of the process. Why is this important? If I had let's say, if I actually had a blue cone that only reacts, let's say, to a 120 wavelength of 120 nanometers, which would be these short waves, right? If I only had that, I would only be able to see blue color. And then I would be only able to see green and red. That would be 3 colors. But that's not what I'm seeing. I'm seeing multitudes of colors because this distribution allows me to have 2 or 3 cones being active at the same time, allows me to see the full spectrum here. So if we had only actually red, blue, and green, we would not be seeing all that we are seeing now, but only those 3 colors. So it's really, in that sense, it's really wrong to say that we have only those. No, we have short, medium, and long ones. And we can also measure the intensity of our rods. They are also here on the list. And when you look at that layer of retina, this is how you find them under very good microscopes. Now, I would like to actually show you something. Can you see it here? I'm sorry I put it down here. I didn't realize what would be the setup here. But can you see this drawing here? Okay, this is left to us by Sir Isaac Newton back in 1704. His representation of color. Now, this is what always baffles me, is how these geniuses come to the conclusions, to the correct conclusions, some 250 years before the rest of us. He's showing here, and this is in his handwriting, he's showing here his representation of color, and he actually has this part of the spectrum shown differently from the rest of everything. So this is Sir Isaac Newton, the same one who back in 1966, when the flag was all over England, left Cambridge, went home, went to the fair, bought the prism, played with the light and prism. I'm sure you all know the story, right? And he saw the light and the rainbow. So he knew how the rainbow looks. Still, he draws this circle. Do you have any idea why? Some of these colors are separate. Huh?
Invisible.
Invisible. Well, you can see it.
This part.
But can you see that part?
Normally, no.
You cannot see normally. But what is normally?
Ultra and infrared.
Huh?
Ultraviolet and infrared is not for our spectrum vision. Yes.
Keep thinking, you might be on the right track. So we can see all of these colors, and Isaac saw all of them, but he saw that there is something special with those. Think about it. Think about his experiment. What's so special about these colors?
Only is long as the short distances.
Long and short distances? Okay.
Between waves.
Let me help you. So this is what he saw in his experiment, the rainbow. What is this? Okay, so this does not exist in the rainbow, these colors, but we see them easily. And the fact that we see this color is a courtesy of our biology. So he noticed that there are some colors that he did not find in the light, but you can easily see them. Why? Because sometimes you get this part excited and this part excited, and phenomenologically you get a full circle out of very different physical stimuli. So it's a courtesy of how our biology works. And this is very important for vision and for our brain in general. Things are not quite the way they are. Some things get distorted by our biology. This is an excellent example. So this is how you actually see things. You have 3 types of cones, but in order to see different colors, they are active in different proportions. And so basically this is how the system actually allows you to see some colors that are not even present in the rainbow, or some wavelengths. And then we can see, of course, what happens when you can't see all of these wavelengths. So basically, if you see all of the wavelengths, you see this whole range, and this is how this lovely, colorful picture looks. But let's pretend that a person doesn't have one of these cones. For example, doesn't have short waves. Then the picture starts looking like this. So this is what biology does to you. This is gathering information, but only through biology that you have. Okay, what happens if you don't have middle? And what happens if you don't have short? Long. Okay. So you can immediately spot is that two lower pictures are not that very different. And you already know why, if you look at this graph. Look, these distributions are almost overlapping. So one of them missing is not going to produce that much of a difference, whereas this one missing is going to produce a lot of difference, right? So you need to think about this distribution. That's the first modeling rule here. The next thing is, how do things get projected on the retina? So if you have something like this in the world, and it comes to your eye, it will turn around left and right, and up and down, and that's going to be the projection on your retina. So people back in the day, like Descartes and others, were looking at these projections at the retina and thinking, okay, so this image is inverted, so somewhere in the brain must be a process that re-inverts this in order for us to see things not upside down and so forth. However, you are not to think about this as an image. You have to think about this as an activation of those layers of cells. There is no picture here. There is no image here. Okay, this is just an activation, and from now on, these are just activation of the cells. And I'd like to stop here and talk to the people about this. Don't imagine little pictures of the world in your brain and little movies. Because when you talk about vision, because we are such a visual people, all of us, it is easy to imagine these pictures and these movies. And even if you use your next best sense, hearing, it's easy to imagine little, well, songs in your head or voices. But then you start talking about other senses. Smell. Do you have little perfumes in your head? Taste. Do you have little cakes, bacon pieces in your head? Well, you don't have pictures, just like you don't have little perfumes in your head. All you have in your head are neural impulses that actually represent the world outside. But now, since I actually killed that lovely magic for you, I should give you some real magic and tell you that, in fact, by the way eyes are positioned in the head, you can tell about organisms' lifestyle. So if you look at this, for example, here is a good example of a hunter. What does hunter have? Eyes in front. So there is a huge overlap of visual fields. Give you a very good depth perception. And here is a for prey. What does prey have? Eyes on the side, so covers a huge view, but no good depth perception. Whatever moves, the prey doesn't know how far that is, just runs, right? So that's the difference. So where do you think you are on this continuum? Huh? Well, there you are.
On the right.
Well, we have actually quite a decent overlap. Quite a decent overlap, but also we can see about 320 degrees, but nothing like this. This rabbit can see behind himself. Yes. Okay. And so this is how it looks when we go further. And so visual field, left and right, then project. And when things come from retina, further in the brain, some of the information switch to the other side of the brain. This is actually biologically extremely clever mechanism, because for an animal, most likely thing to happen is, if it leads certain kind of life, for example, is a prey or a predator, is maybe to lose an eye. If you lose an eye and stop feeding the brain, then the brain starts dying and then horrible things happen to the organism. But if you have this crossover of some information, then even if you lose an eye, then both sides of the brain will be kept feeding, fed information, and everything inside stays intact. Extremely clever and a robust solution for how this should work. Now, so you go to these areas, and the first area in a human brain, now this is human brain, or rather model of a human brain, you get visual area 1. That would be the pink. And it's located here at the back of your brain, and this is, let's say, left hemisphere. And then you have a visual area 2 and visual area 3, and so forth. This is how it works in the brain. And the lovely thing about the brains are, this is when usually when I talk about these, people are like, yeah, but is this for everybody like that? Yes, it is for everybody like that. Each one of us has a visual areas right here. There is no randomity in this. It is always like that. Now I see there is no randomity and there is a system, but it doesn't mean there is no variation. Our brains are much like our faces. They're all the same and all different. They're all the same in a sense, we all have nose in the middle of the face, and then the line that connects our eyes is also almost perpendicular, but it can vary a bit. And then you have about 6.5 centimeters between the eyes, which in reality means between 6.3 and 6.7, and so forth. And then you can have your eyes blue, green, or brown, but not orange or yellow. So some variations are allowed and some are not. And it is absolutely the same thing with the brain. So if you measure the distance, let's say between a cheekbone and a nose for everybody here, you will get some variation. But there is only so much it can vary. And the same thing goes for brain. Here are 3 brains of 3 people. This is how much it varies. Okay. So as I said, just like our faces. Obviously, the fact that somebody has a larger area V1 doesn't mean they see better. Just like somebody who has a bigger nose doesn't breathe better than the rest of us, right? So it doesn't work quite like that. It is just a biological variation. And now, these are just the first 3 areas. And what I'm going to show you now is an extremely simplified 2D version of brain areas from 25 years ago. You can see V1, V2, V3, V4, and the rest of the areas. As I said, this is extremely simplified 2D version from a long, long time ago. And then we do more research to find out about these areas and what do they model. Now, the question is, why do we need all this brain power to actually compute this percept? What do they do? Well, once the light came in, triggered our photoreceptors, and the information went from retina through thalamus, through visual area 1, it starts actually resolving the information about the world around us, about shapes and locations of things, about illumination and shadows in the field, about the color in the field, and about the textures. So this is the information that came through light about the objects light reflected on, and now our visual system has a task to somehow get this information back. And the way we are trying to find out how this visual information is actually obtained, we do experiments. Here is one experiment in which you take students, show them the movie they never heard of, and you actually measure their brain activity. Okay. And then you get something like this. So this research is done halfway between that scheme from 1991 that I showed you, and today it was done some 12 years ago. And it has areas that are still not put in that scheme I showed you from 1991. These are the areas that, when we have discrete pictures, so not yet a movie, but we are just starting, get lightened up when we show these people red faces, blue objects, like these objects, and green buildings. And when we turn on the movie, then it gets much nicer. Yes? So this is what you do in the research. So this is just to tell you what area, so the spatial representations of this area, but what is really happening during the vision? Well, this is when you need a timeline. So this is the time in the movie. 36 seconds, 96 seconds in the movie, 186 seconds, and so forth. You measure everything that happens during the 2 hours of the movie. And then you have certain situations in time when the area you are recording from is exceptionally active. So you go back to your movie and you find what is happening when that area is active. So when this area is active, this is happening. When this area is again extremely excitated, this is happening. And again, here we are. So we dub this face area, because this area always gets exceptionally activated when you see a face on the movie. Okay, how about this? The reports from the brain area now. So this area gets very excited in some other times when we have scenes. So okay, here we think, well, you would guess that faces of other people are important to us, scenes because we need to perceive the space around us, that is also very important. But then you keep researching and recording from our other areas, and you end up with the things completely unexpected back then in 2004. For example, there are areas that get exceptionally excited when you have hands. When there are some actions. Turned out, after a research from Professor Isolati and his group in Italy after this, that there is an area in our brain exceptionally interested in motor actions that can be done with our own bodies. So not if I leave this to drop down, that is not going to activate your area. But as long as I'm waving my hands, this area of yours in your brain is going to be exceptionally activated. Why is that? Because it seems that this is helping us in social perception and joint actions. So when we want to carry something together, we need these areas. When I'm actually in a street fight, by the size of my own hands, I know how far I need to be from my opponent in order to be unreachable. When I'm dancing with somebody, I know how close I need to be. So this area actually reacts to what other people can do, and I can do as well. And through my own body and the representation of my own body, codes the representation of other bodies that I can be in touch with or in contact with. So this is just one of the things that we learned. This is back in 2004. This was in 1991. More like today, latest news on how these brain areas look, 2012. Now we try to represent them in this way. Now this is no longer silly two-dimensional map, as you can see, but this is actually trying to represent the brain the way it works. So maybe some of you were interested in the problem of how to represent Earth, but you do two-dimensional maps from the sphere. That's simple. Brain is much different. And when you try to flatten it out, you end up with a much worse representation of how big areas are. So we take this tentatively, some sort of an imprecise roadmap, where is what. And then we try to see, feature by feature, how these things are organized in our brain. So for example, if we take one feature such as color, here we come to my second trick question for you. Is green a primary color? Hmm?
No.
No. Yes. Who says yes? Nobody? Okay, a couple of people who says no. Some people. Are there any artists among you? What do artists say? Probably not. Painters would say no. Of course green is not a primary color. Have you ever been in elementary school and mixed the paints? Yes, exactly. Green equals yellow plus blue paint. People like me have an opposite opinion, but our logic is completely different. Green does not feel like a mixture of any other colors. Orange might feel like a mixture, but green does not. And there are some other classes of people who would agree with me, that's why I'm mentioning them here. You are probably all familiar with this logic, right? How do you get the colors on the screen? So you don't get them this way at all. You get them very different way, and then yellow equals red plus green light. And then you can get all sorts of intensities among these things. So depending what's your logic, green can be or not a primary color. Yeah? Do you agree? I see some. Okay. Anyway, so when we talk about phenomenology, we can talk about chromacity and achromatic colors and divide everything into two classes, and then we talk about several attributes like hue, and then in psychology we think about four primary colors and the complex primary mixtures, and then we distinguish the intensity of the color, let's say blue in this case, and the purity of the color, let's say red in this phenomenological model of color is in 3 dimensions. First one we call hue, and this is the circle I've already shown you when I was talking about Newton. So this is phenomenology, right? So this sort of corresponds to wavelengths, sort of course. And then dimension perpendicular to this hue is lightness, or perceived intensity of the color. And it goes from black all the way to white. And then the third thing I was talking about is how intense or pure color is, and it's a saturation. So this is a third dimension. It starts with very saturated colors, and it ends with gray. This is a 3-dimensional representation of color in our phenomenology. And it corresponds to this physical stimuli. And as we said, every lambda from the spectrum is perceived as different color. In the best case scenario, this would be the, we would have this, but more likely most of humans are somewhere here, right? So most humans, just like you cannot hear every sound, something is too loud, something is not loud enough, so you can't see everything here. And the way we deal with this, what you can see is in experiments. So imagine this kind of experiment where I'm having all sorts of different wavelengths, and I'm measuring the difference my participants can see. So this is how psychological experiment with colors really look like. Obviously, not in a space like this, but in a well-controlled space, I show them something like this. And I tell them, do you see this color? And I know exactly where this wavelength is, right? So I know exactly where this wavelength is. And I will measure the difference that my participant can observe. So I change this slightly, and I change it for 1 nanometer. Of course, I'm exaggerating things here so you can see what I'm doing. But in reality, I change just 1 nanometer, and I ask my participant, do you see the difference, or is this whole circle the same? And let's say here she says it's the same. So I said, okay, so I change yet again for yet another nanometer. So 1, now we change for 2, right? And I say, well, do you see the difference now? She says no. So I add another nanometer of change. Do you see the difference now? Now she's happy. She says yes, the 2 halves are different shade of yellow. Excellent. So I say, okay, somewhere in this, here in the spectrum, the difference is 3. So then I take this color, make another circle, and then I repeat. Do you see the difference or not? And I do this across the whole spectrum, and then I end up with my results. And as you can see, in some parts of the spectrum, 1 nanometer change is already enough to see difference, but in some parts of the spectrum, we need to go all the way to 5 or 6 nanometers to see the difference. So, how many colors do you think people see in this way, in these controlled experiments? Huh? Well, do you have any idea?
How many?
How many different colors then people end up seeing? 1,000. Now come on, dear. You can't see 1,000 between 120 and 700. You can't see more than 300, obviously. So, okay, when we know that on average 2 or 3 is the difference, then we end up with 128 hues that people can see in these kind of experiments. But remember, I'm talking only about one of the 3 dimensions, not about everything. This is just a hue. When I add all of these dimensions, then we're much closer to what you initially suspect. We can see a lot of different. This is the best thing I found, and it only is really good pixel by pixel on some monitors. Obviously not on my laptop, not here, but actually there are all these differences that you can see. The human eye can distinguish about 10 million different, yes, you were right, but 3D colors, not just hues. But hues times saturations times lightness. Then you end up with this number. Okay? And then we have different way to represent them, more, and you found this, and some of these, see, some of these things look very pixelized, and some of these things are not so pixelized. That's because you are very, very versed to distinguish in this part of the spectrum, right? So wherever you can see pixelized things, that's where your sensitivity is quite good. Wherever it looks more flat, then actually it is your sensitivity that is worse. Obviously, I have not tested this slide projector and so forth, but most likely it is your eye. So back to my trick question: is green a primary color? Well, it depends whether you are doing mixing like the artists or like the engineers. So if you're mixing the pigments, the more pigments you add, you end up with that, well, mud color. You probably mix pigments sometimes in your life, and the more you add to the mixture, the darker and more horrible it becomes. Obviously, this is coming from object surface. This is mixing of the pigment, and it is partially absorbed. So more pigment you add, the more different wavelengths are absorbed, and you end up with a dark color. On the other hand, if you have mixing of the lights, it's additive. So the more different light you add, you end up with a white light in the end, right? Just like Newton's experiment had showed us centuries ago. So this is what is happening also on human retina, because what happened, the mixture we have is actually mixture of the lights, not mixture of the pigments, and the lights are added. So if you look at the middle of this, you would know whether it's subtractive or additive process, right? Whether you end up with white or the black. So this color mixing, when it comes to computers, and I'm going to, I guess, just remind most of you, because you probably know these things, we have this color model. And curved ring here will give you spectral colors, whereas this bit, straight border, are those extra spectral colors. Those are those that Newton noticed that you don't have in the spectrum, but we can still see them, right? And of course, then you have this cross, which is an achromatic center. That would be in a three-dimensional space where lightness hit the hue, okay? And then you have the primaries. And then this triangle among primaries will actually determine what your device can represent. So when you want to make your devices, computer screen, TV screen, and so forth, better, what you need is to stretch these primaries closer to the full spectrum that humans can see, and that's how you make your device better. And whatever falls into the triangle will be what your device can represent. Okay, so this triangle represents screen colors. Okay, and now in the phenomenology, this all looks a tad different. So we have a famous case of simultaneous consciousness. When I show you the two orange targets, shown just by themselves, are they orange? They look exactly the same. I hope you agree. But as soon as I start playing with them, and even simple play, like putting them in the context, they will start looking different. So this is not a change of representation on your screen, on your beam. Again, just like with pink colors, this is a courtesy of your biology, that you see things like that. So our visual system doesn't take just the target, but the whole scene into account. So percept change because I have changed something around the color. Now I would like you to stare at this cross. Now put down your cameras. Just stare at this cross, not look at me. Stare at this cross, not at me. Cross, please. Can you please look at the cross here? Thank you. So don't move, don't laugh. Just look at the cross. I need you to actually look for 20 seconds while I'm telling you things. And I promise you a very nice effect. Just don't move your head. Just keep staring. No, don't move your head. Just keep staring, motionless, into the cross. And I really promise you a very good effect. Once, 20 seconds of it. Don't talk. That moves your eyes. Please don't talk. Just stare at the cross until I do this. Can you see that? Woo! Now, if you were a good kid and stared at the cross as I asked you to, you saw something very pretty. Yeah? Yeah? around, the effect is extremely fast. Yeah? Because once your eyes get accustomed to it. Okay, so this is called color after effects, and these are, again, the biology. So the longer you activate green cell, the moment it stops being activated, and you show the white light, it shows into yellow, it goes into opposite color, pale complementary colors. And you have a lot of these lovely effects that are just a question of how our brain gets the information. I love this one. So these are, you hopefully agree with me, identical gray targets. Now, obviously, I'll start with the magic. If you observe this one, that's where the most action will happen. When I start doing this is changing their background. Okay? Now, compare the first and the last circle. Identical circle. Now, what I like to show you at this point is how little your brain and knowledge has to do with the things. Let's try again. Now you know everything. Does it help you? Can you see them as the same? Try hard. But you can't. Even I who make this, I can't do that. Yeah? So your brain is actually completely powerless when you consider the way information is being picked up by a visual system. So this is all important when you go for modeling. This really looks different no matter what you know about the things. But we really need to know how come something like that changed the appearance very much. There are additional phenomena happening around here when the colors get very similar to target, but I won't bother you, but there are a lot of lovely experiments on that as well. When we talk about what happens with colors and why they change, we talk about constancy of the color, the fact that colors look the same in the changing conditions. And there are two of these conditions that we talk about. So we have two gray targets, and the first difference is illumination. So, basically, you walked in with your stuff, your white dresses, blue pants, brown bags, and so forth, from the daylight, which is much more intense and different in wavelengths than the light in this room. Still, all your clothes or all your stuff looks exactly the same color as outside. Meanwhile, what I did with the background, just changed the background on one of the targets, all of a sudden it looks completely different. Now that is a problem. How does this happen? This problem with illumination we know. What do you see here? Do you see red roofs, green trees, yellow walls, right? And then I take the same picture in a very different illumination. Now this tree is really green, but before you didn't have a problem calling it green, right? So it really does change a lot, but you are completely oblivious to this illumination change. And this is basically a number of photons emitted at different wavelengths. And perceived range of this illuminance for us goes to between 1 and 1 million. So it can be a really powerful range. Remember I said 1 quantum of light. And this is the range. So we are talking about extremely well-developed, sophisticated sense. And it tells you this is a green tree, no matter that light change. You can recognize the objects. Which means you go and park your car at the parking lot in the day, you come out of your lobby at night, you recognize your car. It's red or white or yellow, just as when you parked it this morning. There is no difference. Meanwhile, I show you the different backgrounds and you get confused. Now, every surface reflects a certain amount of light. If it's black surface, it reflects 3% of light, white surface reflects 90% of the light, and everything else is somewhere in between. And then you have light sources that can be powerful light sources, and once the light shines on the surface, it gets reflected 3% or 90%. Or it can be very different light sources, not so powerful, but still you get 3% reflectance as opposed to 90% of reflectance. And what is reflected is called luminance. And this perceived luminance, in perception we call brightness, whereas this perceived reflectance we call the lightness. Now, what we want to know when we model vision is, how come we see lightness? How come we see true color of things, never mind whether we are under one or the other illumination level? Because this is how we recognize our stuff. They have certain color, they have certain shape, they have certain size. I don't imagine that people here are this size and people in the back are that size. I know you are everybody more or less the same size. I don't imagine the people in the first row under the window have bright colors and people in the back do not. When I look things from different point of view, I don't imagine they change shape. I know they don't. So there is this constancy, despite the ever-changing visual conditions, that my cognitive system is constantly sorting out. And this is what we really try to model, and this is really our task. How come you constantly, online, are picking up this information, which is somehow meeting whatever you have in your memory, and comes up with the right combination, which is to say, aha, this is mine. I know this by color, shape, location. But if you change a shape or a color or location, but not too much, you can still recognize it, right? So it gives me some kind of variability. Of course, when I try to model this like physicists did in the beginning, so this luminance is obtained this way. It's reflectance time illumination. But if I model perception, then I'm here with a problem. Luminance is the thing that reaches with the eye, whereas these two things are unknown. So when you first walk into this room Monday morning, you didn't know illumination level and you didn't know the color of these chairs. They did not confuse your visual system at all, right? Immediately you knew some are blue and some are gray and some are black, right? You were immediately aware of this information. And this is the equation with two unknowns. So the first idea people had in the 19th century is, well, if you look at many, many things around here and you put a lot of equations, you end up with a system, you solve the system, and that's all lovely. Okay, so you try that, but unfortunately you're always lacking one equation to solve the system. So you can't. You need something else. And then we look at these things, the situations we actually have in the vision, and say, okay, well, what is the case here? Our L's are the same in this situation, right? And in this situation, it is the same L, but in this situation, it's not, because you have a shadow, right? But what happens with the percepts when you ask people? Well, here they're more or less the same. The tree is green, right? And there are quite different. So what is lacking is something else in the equation that can come from this L to this P. The question is, what is that is lacking? Do you follow the question? Okay, so this is the thing that we will be talking about at the workshop, how to create the illusions. So modeling of the perception actually let us create most wonderful illusions because we know where the system is doing something strange. For example, what has changed here but has not here is ratio to the background. So what is lacking to get to this equation is a ratio to the background. Ratio here and here changed. Ratio here and here did not change. So these are some of the simpler things that I can demonstrate you fast. But of course, as we go through the system, it gets more and more complex. But however, if you know how it works, as I said, you can create very nice illusions. Again, here I am with the two identical gray targets. And this is created by our colleague Dejan Todorovic, who knows a lot about visual perception, who can create these lovely illusions. And he shows you these identical gray targets, and then gives you a wider view, and then a wider view, and then a wider view, and then you see the whole thing. Those surfaces look really, really, really different. And again, no knowledge can help you, right? So this is purely visual phenomenon. Okay. This is combined knowledge about background and illumination effects. Okay, well, thank you very much. Please.
But they just don't know how to put it into picture.
Actually,
Why?
we are very used to doing this, but losing one dimension in order to represent things is extremely artificial process. It needs to be acquired through extremely specified experience, and it doesn't come naturally. And we know this from work with children, we know this from work with tribes that live in remote locations. This really does not come naturally. Actually, we know this as well in the science, where we always, because we are always inspired by mathematical models, and in the end we try to describe everything we do through mathematical models, because only once we have mathematical description we have an idea that we understand things. And then, for us, following mathematical logic, most simple thing is a dot, and then you have a line, and then you have a plane, and then you have three-dimensional space, and so forth, right? However, it doesn't necessarily work like that with biological system. For a biological system like ours, 3 dimensions is simpler than 2 dimensions. 3 dimensions is what we evolved for. 2 dimensions is not what we evolved for. We invented this. And we are quite good, turns out. But we are good in many things. We are good in, you probably saw sometimes Tom and Jerry cartoons. And you flap Jerry, and he becomes 2-dimensional, and pop, he goes back into 3 dimensions. And our kids can understand what's going on at the age of 3. The only thing they can't understand is this is not realistic. But they can understand what's happening. Somehow, our cognitive system, our brains, allows us for way more than what physical reality had to offer. And it's quite a mystery. Why can we pilot the plane that goes 1,000 kilometers an hour when we are actually developed for the speeds about, well, what, 30 kilometers an hour? What's the fastest human being running? What, 60 kilometers an hour? How much? I don't know the... 42. Kilometer an hour. That's the fastest human being, right. And then there is our pilot doing it nicely 1,000 kilometers an hour. That is all of you doing it 120 on the motorway, right? That's not what you evolved for, but you're very good. So we can do apparently all of these things, but not all of them come naturally to us. So you have to, I mean, when you have students, you actually have to keep this in mind. 3D is simpler than 2D, unlike math taught us. In biological system, the dot is not the simplest thing. Okay, thank you. Any more questions?